Bismillahir Rahmanir Rahim. In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. Dear students, I welcome you in Pakistan International School Types virtual learning class. And this is biology class for grade first year. Dear students, in today's lecture number 29, we are going to continue our chapter number 4, bioenergetics. And in bioenergetics, we are going to start another new topic of this chapter, that is cellular respiration. And in that, we will see different types of cellular respirations, that is anaerobic and aerobic respiration. And in aerobic respiration, we will see glycolysis. So let's get started. Dear students, you know that cellular respiration is very different from breathing process. Breathing is just take in and take out of oxygen and carbon dioxide, but in cellular respiration, the breakdown of glucose is done in the presence of oxygen. So it is a catabolic, exergonic, oxygen requiring process that uses energy and extracted from macromolecules, which is from glucose, and to produce energy and water. So the overall reaction of cellular respiration is glucose in the presence of oxygen. We get carbon dioxide and water, and the main purpose of cellular respiration is to provide energy in the form of ATP, and that ATP is utilized for different body processes, different regular activities of the body. So cellular respiration, we know that that is oxidative reduction is a chemical reaction that involves removal of hydrogen atom from one molecule and gain of hydrogen atom to another molecule. Like we know that cellular respiration is oxidation and reduction process. So now we will see what are different kinds of cellular respiration. Actually, for cellular respiration, we need a certain substance just to utilize it. That is glucose. And that glucose acts as a respiratory fuel, because that is actually, we can also say that cellular respiration is a burning of a fuel in the form of glucose, by which we get energy. So here we can say that respiration or cellular respiration is having different types. Like if we see that cellular respiration just starts from glucose, the breakdown of glucose in the process that is called glycolysis. Glyco means glucose, lysis means breakdown. So in the form breakdown of glucose to pyruvate. So this all is happened in cytoplasm. Okay, the conversion of pyruvate up to pyruvate. But conversion of pyruvate up to pyruvate, we just get 2 ATPs. And if we, this is not oxidative because oxidation is not utilized here. Oxidation, if there oxygen is present, so we will get through aerobic respiration. If oxygen is not available, so we go to anaerobic respiration. N means without, aerobic means oxygen. So an aerobic respiration is further classified into two types. Like Pyruvate then changes into two um, like categories of we can say the breaking down of pyruvate molecule, that is called fermentation if it is in the absence of oxygen. That is lactic acid fermentation, and then we said it uh, alcoholic fermentation. In that we'll get alcohol and carbon dioxide. If oxygen is present, so pyruvate goes through aerobic respiration, and that is then further changes, or that pyruvate moves into Krebs cycle, then it moves into electron transport chain. So we will get ATP from these, and especially from electron transport chain. So in a pyruvate, like in fermentation, we'll get only 2 ATPs. And in aerobic respiration, or at the end of this electron transport chain, we get 36 ATPs. In eukaryotes, like in human beings, in plants, and in prokaryotes, like in bacteria, we get 38 ATPs. Okay, so we'll talk about mechanism of anaerobic respiration, that how this respiration is going on. So in the absence of oxygen, when the glucose is broken down further in glucose is broken down into pyruvate, and then from pyruvate it is broken down into further products, that is called fermentation. And there are two types of fermentation, as we have discussed before. There's lactic acid fermentation. Actually, in glycolysis, it consists of glycolysis. So glycolysis is the only step which is going to be continued in aerobic as well as in anaerobic respiration. So if in aerobic it goes through fermentation further, but if in aerobic condition where the oxygen is present, so it goes through the Krebs cycle. So here we are going to discuss fermentation like without oxygen. So glycolysis will be followed by reduction of pyruvate by if you see this reaction of lactic acid fermentation, so glucose change into pyruvate, and this pyruvate is gonna change just form or product into lactate or lactic acid. So glucose is 6 carbon compound, 6 carbon compounds. Pyruvate is 3 carbon compound. So we'll get 2 molecules of pyruvate, then 2 molecules of pyruvate are further broken down into 2 molecules of lactic acids. Like it is also having 1, 2, 3 carbon atoms. So glucose, when it during the process of glycolysis, it is like going to be phosphorylate ADP into ATP. So 2 molecules of ADP are utilized, not utilized actually, that we can say that are phosphorylated, and phosphate is added into 2 ADPs. So we'll get 2 ATPs. So at the end, we'll get 2 molecules of pyruvate or pyruvic acid. Then this pyruvate will again transform into lactic acid, 2 molecules of lactic acid. So during the process of glycolysis to formation of pyruvate or pyruvic acid, so this NAD is reduced into NADH and plus 2H. Like it gets 2 hydrogen atoms from this glucose and then changes into NADH plus 2H2, sorry, 2H plus, like 2 proton. It accepts electrons from the glycol, like from glucose, and just change into NADH in the reduced form. So here is a reduction process So after pyruvate, if oxygen is present, oxygen is present, so then this pyruvate will uh, this NAD gives the electrons to oxygen. Oxygen is the final electron acceptor in electron transport chain. So this NADH and plus 2 H, so that gives these electrons to oxygen, and then we will get more ATPs. And also this proton is utilized for the formation of water or production of water at the end of the reaction. But here no oxygen is present, so this NADH will again oxidize. So this is oxidation reaction, and we'll get again NAD plus. So this NAD and NADH reduction and oxidation process will move on again and again. So this is its overall reaction, that this is glucose molecule, then NADH will be reduced into NADH plus 2H, and this is pyruvate. Then this pyruvate is like changed into lactic acid, and this 2NADH and plus 2H, that will be again oxidized in the form of NAD plus. Let's talk about alcoholic fermentation. In alcoholic fermentation, you can see in this step, the same process up to pyruvic acid is the same, like glucose is changed through glycolysis into 2-pyruvate or 2-pyruvic acid molecules by the phosphorylation of ADP into ATP, and we get 2 atoms of ATP, or 2 molecules of highly energy compound that is utilized for further physical activities. This pyruvate in alcoholic fermentation, it goes into further like process in which there is pyruvate decarboxylate. It means decarboxylase means removal of carbon dioxide is happened. So removal of carbon dioxide is done here. By removal of carbon dioxide, this pyruvate is changed into 2 carbon compound that is called acetaldehyde. From glucose we get 2 pyruvates. So from 2 pyruvates we get 2 acetaldehyde, and we get 2 carbon dioxide. So here it is a 3 carbon compound. So by breaking down of pyruvate, we'll get 2 carbon compound, that is acetaldehyde, and we get 2 more molecules of carbon dioxide which are removed in this reaction. So the same way NAD gets reduced into NADH, but there is no electron acceptor at the end in the form of oxygen. So this NADH is again oxidized and a form of like remove the electrons and just change into NAD plus, and that again can be transformed into NADH in the reaction. So this acetaldehyde is actually the intermediate product, that is not actually the final product. Acetaldehyde then change into ethanol by adding hydrogen, this one, okay, and we'll get ethanol, 2 molecules of ethanol. So this is our final product. Ethanol is having 2 carbon compounds. We can because OH functional group is present with that, and this way is called ethanol, like it is alcohol, so that's why it's called alcoholic fermentation. So if we see its overall reaction, so we'll get here glucose, then glucose is changed into pyruvate, 2 molecules of pyruvate, and this NAD is going to reduce NADH, where it will be utilized further in if it is aerobic respiration to the formation of ATP in electron transport chain, and this can be utilized for the water production. But here this pyruvate is going to be changed into alcohol, that is ethyl alcohol or ethanol, and this NADH and then again reduced into NAD, and we'll get carbon dioxide also in this reaction. Okay, so if we see the difference between lactic acid and alcoholic fermentation, so we can see that lactic acid fermentation refers to metabolic process in which glucose is converted into lactate and cellular energy in the form of 2-ATP, and in alcoholic we are getting product ethanol and carbon dioxide. Lactic acid fermentation just occurs in lactobacillus, especially bacteria, some yeast, and in muscle cells of human being. When we do lots of exercise, so due to lack of oxygen, our muscle cells starts to produce lactic acid, and due to accumulation of lactic acid in the muscles, we just get muscle fatigue and our muscles get pain. Then that is in alcoholic fermentation that occurs in yeast and some bacteria and microorganisms as well. So lactic acid produces lactic acid fermentation produces lactic acid molecules from pyruvate, and here we get carbon dioxide and ethanol from pyruvate molecule. So lactic acid fermentation involves lactate dehydrogenase, like in which the hydrogen is removed, and decarboxylase as well sometimes. So in alcoholic fermentation, pyruvate decarboxylase is used, in which the carbon dioxide is removed, and dehydrogenase is also used here. Alcohol dehydrogenase is used. Lactic acid fermentation is very, very important for the production of yogurt, yeast, and dairy products, other dairy products which we use from milk, which we make from the milk. And alcoholic fermentation is used for making bakery items like bread, and we also use in brewing industry, like we used to make it produce a beer, wine, vinegar from this alcoholic fermentation. So here we have described about that lactic acid fermentation and alcoholic fermentation. So that is done in human in muscle cells as well, and from alcoholic fermentation we got alcohol, ethyl alcohol and carbon dioxide that used for the production of ethanol. Okay, so dear students, now we will see mechanism of aerobic respiration. Before we have seen a mechanism of anaerobic respiration, and now we'll see aerobic respiration, which is a catabolic process, again in which glucose is used. Glucose is used to break down in the presence of oxygen, and first step of that is glycolysis. Glycolysis is the common process of to make pyruvate from glucose, and that is the common process for aerobic and anaerobic respiration. So let's talk about in detail that how pyruvate is formed or produced by the process of glycolysis. Glyco means glucose and lysis means sugar. So it is actually the breakdown of glucose in the form for pyruvate, and with great we can say usage of lots of enzymes over there, and at the end we are getting ATP as well as NADH, and that NADH is not utilized in anaerobic respiration, but it is utilized further in aerobic respiration for production of ATP. So glycolysis is also said to be called as Embden-Meyerhof pathway. So that was discovered by three German scientists, Embden, Meyerhof, and Parnas. That's what the name is given to that process. And it is essential to all cells to carry out glycolysis for making of pyruvate to get energy. Glycolysis consists of almost 10 reactions which are almost same in all cells and divided in these 10 reactions are divided into five reactions in two steps, two phases. So the first phase is energy requiring phase, in which the ATP which is formed in ATP is utilized, and in second phase ATP is produced. So that's why the first in which the first one is called G3P. The two molecules of G3P are formed. So here the energy ATP is utilized, or energy requiring phase, or that is also called as preparatory phase. And the second one is energy producing phase, that is also called payoff phase or oxidative phase. And we will get at the end pyruvate, then ATP, and NADH. So we have discussed about three fates of pyruvate, like lactic acid fermentation. It can be happen if there is anaerobic respiration and alcoholic fermentation. The third one we are gonna discuss in the next lecture, that is Krebs cycle. So now here we will see glycolysis. Glycolysis, we'll discuss its first phase, that is preparatory phase. So preparatory phase means utilizing of ATP as well. So glucose is phosphorylated by utilizing one ATP, and we'll get ADP, and phosphate is added into glucose, that is at the carbon number 6, so that is called glucose 6 phosphate. So this is the first primary reaction of this whole process. And there is hexokinase is utilized in this reaction. Hexo means 6, so it means at carbon number 6 it adds one phosphate. At number 2, this is actually glucose 6 phosphate formation, that is a reversible reaction. Then we'll get fructose 6 phosphate by isomerization reaction. So we are having first phosphorylation reaction, then we are having isomerization. So in step number 2, for isomerization, phosphohexose isomerase, that is phosphohexose, like six carbons are there and phosphate is added all over there, and isomerase, the enzyme which is gonna change its somehow arrangement, so that is changed into fructose, that is isomers of glucose. So that is the second step, and that is a reversible reaction. The second we can say the second primary reaction, but overall the third reaction, in which again one ATP is utilized. So like again phosphorylation reaction is done, and in which one ATP is utilized and that phosphate is added into fructose 6 phosphate, and then we will get fructose 1,6 bisphosphate, means two phosphates are adding here. And then ATP is changed into ADP. This is also irreversible reaction. Then we will again having isomerization reaction, in which not isomerization, actually fructose 1,6 bisphosphate is going to split now into three carbon compound, because yet fructose is 6 carbon compound. Now you're getting three carbon compounds. One is glyceraldehyde 3 phosphate, that is called G3P, which we have discussed in photosynthesis, and then the dihydroxyacetone phosphate, two molecules of this one. This is one and this is a two. So this is a reversible reaction that it can be changed with another enzyme to fructose 1,6 bis phosphate. So this that is done with the reaction that that is enzyme that is called aldolase. In the presence of aldolase, that is broken down into um, further 3 carbon compound. Up to here, preparatory phase is completed. That is also called investment phase, where we are investing our energy. Now we'll get our energy. So the next process, that's called oxidative phase or payoff phase, from the step number 5. So here G3P is gonna change into glyceraldehyde 3 phosphate will um, mm, we can say that a dihydroxyacetone is changeable into glyceraldehyde 3 phosphate. So we are having a one molecule of glyceraldehyde 3 phosphate here and one molecule of dihydroxyacetone, which can change into G3P again. So now we get, you can see here, through two molecules of G3P. So that can be changed into G3P. So that's why it is reversible reaction. So that we can say isomerization is done, in which that dihydroxy acetone phosphate, that DHAP, that is changed into glyceraldehyde 3 phosphate, that's called G3P, by the enzyme triose phosphate isomerase. Triose because that is 3 carbon compound, and phosphate like changing of the phosphate from its position, and we can say that it is done in the presence of isomerase. Then in payoff phase, now this G3P, that is gonna change into 1,3-bisphosphate, that is we can say dehydrogenase and phosphorylation process. Dehydrogenation means oxidation process is there, and that is phosphorylation, that is also called oxidative phosphorylation, because the oxidation is done here. So 2 phosphate groups are added. So that's why it is 1,3-bisphosphate phosphoglycerate, because these are two molecules. So that's why 2 phosphates are attached with that. One is at the position number 3 on both of the molecules. When it is isomerized, now at position number 1, the phosphate group is added. As well as that is dehydrogenase process also there. Like G3P donates hydrogen to NAD, and hydrogen is removed, and that we can say NAD is reduced into NADH plus H plus. Then we are having very important reaction, that is the first ATP forming a reaction, and that is also called step number 7, oxidative phosphorylation. Sorry, that is called substrate phosphorylation. Okay, so now this substrate is making ATP, that's why this is called substrate phosphorylation. So in that we are using enzyme phosphoglycerate kinase, and now what happened, that ADP is utilizing this phosphorus which is added here in the previous reaction, and now we are not having a 1 here, because at the position number of 1 on both of the molecules we are not having phosphorus, because that is added into 2 ATP, 2 ADP, then it becomes 2 ATP. Now we are getting a first energy molecule here. So next, what happened, this 3-phosphoglycerate is gonna change into 2-phosphoglycerate, like isomerization. Again, this is done in the presence of that is phosphoglycerate. Mutase, like mutation is done, and phosphate is changed from carbon number 3 to carbon number 2, and that is also happening in both of the molecules. Number 9th reaction, we are having dehydration reaction, in which water is removed, and then 2-phosphoglycerate undergoes dehydration, in which the water is removed in the presence of enzyme that is called enolase, and we are getting 2-phosphoglycerate changed into phosphoenol pyruvate. Now, like 2 hydrogen and 1 oxygen is removed from this reaction, from one molecule. Phosphoenol pyruvate in the 10th and last reaction, that is going to change into pyruvate finally in the presence of pyruvate kinase, in which 2 more phosphate are removed, which are added into ADP and changed into ATP, adenosine triphosphate, and that is our second ATP forming reaction. That is also called substrate level reaction. So substrate level reaction is, number 1 is done at number 10 step, and number 2 is at step number 7 and step number 10. So in this all reaction, we consumed 2 ATPs here in preparatory phase, and we are getting a 4. This is 2 molecules of this, 2 molecules of ATP, this one. So 2 we have consumed, and we get 4. So 4 minus 2, overall we are getting overall 2 ATPs in this reaction. So in the previous lactic acid and alcoholic fermentation, we have seen that we got 2 ATPs at the end of the reaction. So energy currency for pyruvate is very, very less as compared to aerobic respiration. If we just compare it with anaerobic respiration, in aerobic we get 2 ATPs, and aerobic reaction we get 34 to 36 ATPs. Yes, dear students, that's it for today. Inshallah, we will see you in the next lecture with the continuation of this topic, that is how pyruvate will enter into mitochondria, because that is done in cytosol, in cytoplasm. So inshallah, we will see Krebs cycle in the next lecture. Wish you all the best. Allah Hafiz.